speaker is Bartolo Favaloro. Bartolo is the head of the laboratory in the Center of Excellence on Aging in the University of Chieti Pescara. His lab focused his work on the role of methionine sulfoxide reductase A and the alteration of redox signaling in cancer cells.
Good morning. Thank you to the organizers to invite me and give me the possibility to show you my work. And excuse me for my voice and for my very bad English, absolutely very bad English. I'll talk about the possible role of the methionine sulfoxide reductase A. The acronym is MsrA in breast cancer. Okay, this is an overview of my presentation. I'll tell you a little about the role of MsrA in prokaryotes. Then we will see the possible function of MsrA in eukaryotic system and in particular in cancer cell models. Then we will see how MsrA could be involved in the redox control of the PI3 kinases pathway. And finally, I would like to share with you our preliminary data about our current research. So, methionine is the only amino acid together cysteine containing the sulfur atom and is very prone to the oxidation by reactive oxygen species. The oxidation of methionine generates two epimeric forms, the R form and S form. The MsrA is involved in the reduction of S form of sulfoxides and is involved in the reduction of free methionine and methionine included in protein. So this redox recycling of methionine could represent a ROS scavenger system, a local ROS scavenger system. And the enzyme could be also involved in the regulation of other proteins that could be regulated by the oxidation of methionine, because MsrA is also able to reduce sulfoxide included in proteins. The catalytic mechanisms involved the thyrodoxine and thyrodoxine reductase system to regenerate the active form of the enzymes. Now, I start to study this protein in bacteria about 20 years ago. It's a long time. I was in Mario Negri Institute. I was involved in a 50 framework program. We have got a project from a European community. And our objective was to identify enzymes involved in the metabolism of xenobiotic compounds. We isolated bacteria that were able to grow on, to use as only carbon source xenobiotic compounds. And my group was involved in the identification of the degradative pathway involved in this metabolism. So we constructed a DNA library from this bacterium and isolated a gene codifying for a classical enzymes involved in the detoxification reaction, the glutathione transferase, and located about 1 kb downstream to this gene, we identify a gene codifying for the methionine sulfoxide reductase. We express the recombinant protein and obtained antibodies to study the function of this enzyme in bacteria during the degradation of xenobiotic compounds. It is interesting to note that in bacteria, this protein is not activated by general stress conditions, for instance, osmotic stress or a strictly oxidative stress, but is activated only during the metabolism of xenobiotic compounds. So when the bacteria were growing in the presence of 4-chlorophenol, a metabolite of a pesticide, 2,4-D, or in the presence of metabolites of the 2,4-D, we obtained an increase in the level of expression of the protein. In addition, the protein in bacteria localize in an environment closest to the periplasmic space of the bacteria, where are located mono or deoxygenases involved in the degradation of xenobiotic compounds. So we hypothesized that this enzyme could protect bacteria by an increase of ROS during the degradation, during the oxidative metabolism of xenobiotic compounds. And indeed, we detect an increase in the level of ROS during the growth of the bacteria at different concentration of 4-chlorophenol that correlate perfectly with an increase in the level of expression of both GST and methionine sulfoxide reductase. So we constructed this scheme that during the growth in the presence of stress of bacteria, we have an increase of ROS, we have an increase in the level of methionine sulfoxide, and MsrA represents an adaptive response to bacteria to this stress and to protect bacteria by reactive oxygen species. About 10 years ago, I moved in an aging research center, and we studied this system in eukaryotic, in particular in cancer cells, because it's known that it's important to control the level of ROS in cancer. ROS represents a kind of biological paradox for cancer cells, because cancer cells have an increase in a higher level of ROS compared to normal cells. And it's important to balance this ROS, because these ROS, as we will see later, are important for the business of cancer. So for cancer cell proliferation, for angiogenesis, for metastatization, as we will see later. But on the other hand, some chemotherapy strategy are based on the increase of ROS in cancer cell to kill cancer cell, the action of adriamycin, the classical action of adriamycin. So we were interested to evaluate whether also in eukaryotic cells and in cancer cells, MsrA could be involved in the modulation of ROS, of reactive oxygen species. So we first analyzed the expression of this protein in several breast cancer cells, and we found that in cancer cells, we have a lower level of mRNA and protein level compared to normal cells. Okay, we studied the gene regulation. I have time. The gene regulation at the promoter level, and identify cis element regions involved in the downregulation of the expression of this gene, but we were not able to identify the transcription factor involved in the downregulation of the expression of MsrA in cancers, in breast cancer cell model. However, our objective is MsrA could control the level of ROS in cancer. So we decided to silence a breast cancer cell line, the MDA231. We selected several stable clones silenced for the expression of MsrA, and we detect in cancer cell an increase in the level of expression of in the ROS production. This increase is not a very high increase. We have about a double concentration of ROS that we verified by microscopy analysis and by FACS analysis. And in addition, we found a general oxidation in the protein obtained from the MsrA silenced cells. Now, the increase in ROS level, in this ROS level in cancer due to the silencing of MsRA is strictly dependent by the silence of the protein. In fact, we didn't find any variation in the expression of the classical system of enzymes involved in the protection by an increase of oxidative stress represented by superoxide, the two forms of superoxide dismutases, the catalysis of or the glutathione peroxidases. But now, what happened to the growth behavior of cancer cells when we silenced the expression of the MsRA? Because in the literature is reported that if in normal cells, primary cells, for instance, melanocytes or retinal pigmental cells, the silencing of MsRA affects the growth of the cells. In cancer cells, we didn't see any variation in the growth condition, but this is true using this method of culture. In this method, the cells are attached to the plastic surface. This is a monolayer method, but the situation changes totally when we simulate a growth condition more similar to physiological growth conditions. So appeared in the last year the importance of the tumor microenvironment, the importance of the stroma and normal cells that are important and support the growth of the cancer cells. When we put our silenced cells in, for instance, embedded in matrigel that simulates a more physiological growth conditions, we found a dramatic increase in the growth of cells. This experiment has been done in a 9-6 wells, but it's also possible to see by only, I say in Italian, occhiometric, by eye, it's possible to see the change of the color of the medium, the phenol red, due to the classical Warburg effect, the acidification of the environment, and we saw this dramatic increase of the cells. This increase of growth is not dependent by a ROS accumulation, but is dependent by a ROS generation here, because we used a DPI, diphenyludione, that is an inhibitor of the NADPH oxidase system. The NOx system is overexpressed in cancer cells and produce superoxide anion that is rapidly converted in the cells in hydrogen peroxides. And we think that hydrogen peroxide is responsible of the redox control. And the phenotype was also reverted by an inhibitor of the P3 kinases pathway, but I'll tell you more in detail later about this aspect. So when we put these cells in nude mice, we saw this very, very dramatic increase in the growth of the tumors. These two falcons contains the tumors collected by the two groups of animals at 48 days after the implantation in nude mice. In addition, the tumors appeared very vascularized, and probably the increase, not probably, the increase of the vascularization is dependent on the expression of vascular endothelial growth factor. We detect a higher level, a much higher level at the mRNA and protein level of the ELRIGfr in the cells silenced for the MsRA. So the silencing of MsRA increase just a little bit the ROS level that are important to support the growth of cancer, but only in 3D dimension and only in physiological conditions. But what happened to the metastatization? We assay this by a test, a classical test of degradation of the matrix, of the degradation of gelatin, and we detect, we detect a higher level of degradation unit in the silenced cells compared to normal cells. So it seems that the silencing of MsrA is important also for the metastatization of cancer cells. And also in this case, we revert the phenotype using the effect of this major invasivity is due to the ROS that are generated in the silent cell. In addition, but this is not known, the increase in metastatization is dependent on the PI3 kinases pathway. In fact, we revert the phenotype blocking the PI3 kinases pathway. Now, enzymes that is involved in the regulation of PI3 kinases pathway and regulate this pathway at the redox level is the phosphatases PTEN. Phosphatases PTEN dephosphorylate the product of the PI3 kinases, the PIP3. We detect in our cells a lower level of PTEN protein in the silenced cells, in the MsrA-silenced cells. We didn't detect any difference at the level of the transcription, at the mRNA level for PTEN. So this lower level of PTEN in the silenced cells so suggests a post-translational modification of the phosphatases. Now, is known by the literature that to control in the redox control of the phosphatases PTEN are involved two cysteine residues, but we try to measure the ratio. There is a simple test to observe the oxidized form of PTEN and the normal PTEN, but we didn't find any difference in the ratio of oxidized PTEN on PTEN in our system. We think that in the destabilization of PTEN, the destabilization of PTEN could be controlled at the level of the oxidation of the methionine, of the sulfur atom of the methionine residues. If we look at the sequence of PTEN, PTEN contains several methionine residues in the lipid binding domain. That is an important domain of PTEN to localize the phosphatase at the membrane level of the cancer, where the PIP3 is produced. And in addition, methionine residues are also located in the active site of the phosphatase. So we hypothesize that the oxidation of the methionine of PTEN could destabilize the protein and could activate the degradation also of the PTEN. We perform a construct of PTEN GST and transfected this construct in silenced cells and mock, and we confirmed the lower level of PTEN GST in silenced MsrA cells. In addition, this gives us the possibility to purify PTEN using a GSH affinity column, and we detect also a lower level of PTEN activity when we express the construct in the MsrA silenced cells. So there are several indications that the redox control could destabilize PTEN at the methionine level. However, a lot of experiments are needed to confirm this. At the moment, we are performing construct with mutagenesis cytospecific to change the methionine. But by the way, we think the direction is a good direction because we detect in primary cells, in fibroblasts obtained from mice knockout for MsrA, we found a lower level of PTEN in these animals. So this scheme summarizes the results that we obtained in cancer cell model that I show you until now. So when we have a downregulation of MsrA, we observed an increase of ROS production. The increase of ROS production, we think that is an increase of hydrogen peroxides. We use the DFCA that is considered a specific for hydrogen peroxides. And obviously, hydrogen peroxides, we see that is dependent on the NOx system, but as I told you before, NOx system doesn't generate hydrogen peroxides, generate superoxide but is rapidly converted in the cell. So MsRA could represent a local control of the redox signaling at the level of for instance, in pathological condition, to destabilize PTEN. As I show you, the sulfoxidation of PTEN could alterate the localization of PTEN in the membrane, but could activate also its degradation. It could affect perhaps also the phosphatases activity of PTEN. This leads to an increase in the product of the PIP3 kinases, the PIP3 that activate the AKT that is well known, activate the cell proliferation and survival signaling in cancer. Now, this data in the cellular model has been a good indication that the direction is ready, comes by the fact that we analyzed the expression of the MsRA in breast cancer tissue. We used a plate, a commercial plate, where are spotted cDNA obtained from normal tissue and from cancer cases. And we detect a lower level of expression in cancer cases compared to normal tissue. In addition, we organized this sample on the basis of the classification of the tumor classification in tumor more aggressive of grade 3 and tumor less aggressive of grade 1, 2. And we detect a lower level of expression of MsRA in more aggressive tumor than in tumor less aggressive or type 1, 2. So this is an indication that MsRA could the downregulation of mRNA and this redox signaling could support the growth of the tumor, but also could be involved also in the metastatization process of the tumors. Now, I would like to share with you some preliminary data that we obtained with studies in mice knockout for MsRA. We have two models of pathology at the liver, also because liver represents the tissue that have the major expression of MsRA. And we have two pathological contexts, the steatosis that we obtained with a diet without methionine and choline. And after 2 weeks, we have a condition of steatosis. By the way, at the moment, I can't tell you that MsRA is involved in the steatosis. This could be only a stochastic event of this image, because it seems that we have a major lipids included in the hepatocytes. But I have not the statistical data to confirm this. But I have the statistical data from the pathologist that give us the information that in the liver, in this model, after the steatosis induction, we have a more inflammation focali in the liver from the knockout animals. Now, we know that we also see in the before presentation that an important role in inflammation is played by macrophages. Now, at the moment, we are looking at whether in macrophages, MsRA could be involved in the regulation of ROS. And we found that when the macrophages obtained from the bone marrow of the animals are activated in M1 population, we observe an increase in the production of ROS in the macrophages obtained from the MsRA knockout animals. So this suggests that also at the physiological level, MsRA could control the ROS production in macrophages that are important to trigger inflammation conditions. This this indication is also suggested by the other model of pathology that we have, and a fibrotic model that we obtained treating the animals with carbon tetrachloride for 4 months. And as you can see, we have an increase in the damage, in the liver damage, due to the fibrosis conditions, and we detected in MsRA knockout animal an increase in the level of expression of collagen type 1 that is responsible to generate the fibrosis conditions at the liver level. So the conclusions are that MsRA could play a role in possible to could support the growth in cancer, could be involved in the metastatization, but could also regulate particular function, also physiological function, at the level of the PI3 kinases, for instance, in macrophages and inflammation. So I need to thank. We are a very, very small lab. We are only 3 people, 3 persons, work with me. I am a researcher, work with me another researcher, that is Antonella De Luca, Cristina Bichanova, that came from Prague, and I need to thank Antonio Tamburro, that worked with me in Mario Negri Institute during the European Community Project, Herman A. Pieper, that was the coordinator of the project when I studied bacteria, Fabio Sanna, that is a postdoc that contributed to obtain several data I show now, but now he works in Oxford, Rossano Latanzio, that is a pathologist of the center, and Cosmo Rossi, that is the boia, no? Not boia, is the responsible of the animal care, sorry. Thank you.
Questions?
Please?
Do the knockout mice you described just, you mentioned, do they have any overt phenotype? Is it total knockout or is it tissue-specific knockout you use?
Sorry, I don't understand.
The knockout
if I see the animals are normal, have no problem but we think that when we create a knockout animals, if the animals have no problem, we have a lot of changement in the gene regulation and compensation effect that, but we have no specific phenotype. It's been described in the literature that during the aging, animals knocked have a lower expectancy of life, but this was not confirmed by another paper, but probably could be involved the alimentation also of the animal.
And it's a total knockout you have?
Please?
It's a total knockout.
Is?
Knockout total. It's not tissue specific.
Tissue specific? No. This is not tissue specific. No, this is a good question. Yes.
Is it? Are these
no, it's a
flock?
knockout that we is from Jackson. No, totally.
Okay. Thank you.
I have a question concerning the therapeutic approach that you imagine in the future. It is possible to obtain activators of MsRA?
Allora, so we have problem for the activity of these, to measure the activity of these enzymes. Because there are several methods that require HPLC analysis. There is no a simple method to measure the activity of the MsRA. We think that this could have a good prognostic value to determine, for instance, in the prognostic value in cancer cells. For instance, we had, but at the moment we have only a few cases, for instance, from surgical, where we have a very, very small breast cancer that usually are considered in T1, no dangerous breast cancer, but we detect no expression level of MsrA, but this cancer have already lymphnodal invasion. So could have a prognostic role, but obviously we need to increase the cases.
Thank you. If there are not more questions, we can conclude this section. Thank you very much for your attention, and do not forget to visit the poster section and to visit also sponsor and to complete the quiz for this afternoon. Thank you very much.